Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Imagine this. We've been battling cancer with these incredible immunotherapies, right? Celebrating amazing breakthroughs. Yet for, well, a surprising number of patients, these treatments, our best hope, they don't work. They don't work. What if the reason, the resistance, isn't just in the tumor cells or even the immune system we're trying to wake up, but somewhere we rarely look? The nerves. Today, we are taking a deep dive into a really groundbreaking article in Nature. It's called Cancer-Induced Nerve Injury Promotes Resistance to Anti-PD-1 Therapy. Our mission here is to figure out how cancer cells actually deliberately injure nearby nerves and how this interaction, this kind of neural sabotage, drives resistance to powerful anti-PD-1 treatments. This isn't just, another paper. It feels like a real shift in how we think about cancer. It offers fresh hope, maybe totally new strategies for patients who aren't responding right now. So get ready for some serious aha moments. This might just flip your understanding of cancer biology.
It really is an exciting area. And you're right, anti-PD-1 therapy has been revolutionary in oncology. It's given patients years, sometimes decades they wouldn't have had otherwise. But yeah, despite the successes, there's this significant chunk of patients, maybe 30%, maybe up to 70% in some cancers, who just don't respond. Or they respond initially, but then develop resistance. And tackling that non-response is, frankly, one of the biggest challenges in cancer research today. For a long time, we've really focused on the tumor cells, and the immune cells immediately around them. We knew about something called perineural invasion, PNI. That's where you actually see cancer cells growing along nerve fibers. And it's always been a bad sign prognostically. Usually means a more aggressive tumor, worse outcomes for the patient. But why? Why did PNI make cancers nastier, especially regarding the immune response? That was kind of a mystery. We mostly thought of it as just a symptom of advanced disease, not really a cause of problems. So what this deep dive is all about, and what's so paradigm shifting, is this idea of cancer-induced nerve injury, or CNI. The core idea is that tumors aren't just sort of passively bumping into nerves, they're actively injuring them. And crucially, that injury then feeds back and shapes the local immunity, essentially creating an immunosuppressive shield that blocks anti-PD-1 therapy. It really pulls neurobiology right into the heart of cancer immunology in a way we hadn't fully grasped before.
Okay, so it sounds like they didn't just jump into lab models. They had a feeling this nerve connection was real from the clinic. What was the patient data that first hinted nerve injury might be putting the brakes on anti-PD-1 therapy?
Absolutely. They started right there with the real-world impact on patients. They looked at data from 56 people with cutaneous squamous cell carcinoma, that's CSCC, pretty common skin cancer, who were getting anti-PD-1 treatment. And the results were, well, stark, a real wake-up call. Patients who unfortunately didn't respond to the therapy had a much, much higher rate of this PNI. We're talking 50% of non-responders showed nerves infiltrated by cancer.
50%.
Compared to only 15% of the patients who did respond well.
Wow, that's a huge difference.
Exactly. A threefold difference. It's like a massive red flag waving from the nervous system.
Yeah.
And it wasn't just the physical invasion. These non-responders also had higher levels of a specific nerve distress signal, a marker called ATF3 in their nerve tissues.
Oh.
You can think of ATF3 as like the nerve cell hitting its panic button, signaling it's under serious stress, serious injury. So yeah, very clear correlation right there in human patients.
That is powerful. But CSCC is just one cancer type. Did this connection hold up elsewhere, or was it specific to skin cancer?
Good question.
Yeah.
And no, it wasn't isolated. That's key. To see if this was a broader thing, they developed what they called a PNI nerve injury gene signature. Think of it like a molecular fingerprint for this nerve damage pattern. They applied this signature to much bigger data sets from patients with other really tough cancers, metastatic melanoma, gastric cancer. And the same pattern popped up again. This nerve injury signature was consistently higher, enriched, in the non-responders to anti-PD-1 therapy across these very different cancers. It strongly suggests this is a tumor-agnostic effect, meaning it's probably not just about CSCC. It could be a fundamental way various cancers evade treatment by messing with nerves.
Okay, so the patient data is compelling. Strong correlation. But like you said, correlation isn't causation. This is where it gets really interesting for me. How do they actually prove it? Prove that the nerve injury was driving the resistance, not just tagging along for the ride.
Right, this is where the elegant mouse work comes in. They designed some clever experiments to get at causality. First, they did what's called a denervation experiment. So they had tumors growing in mice, but they surgically removed the nerve supplying that specific tumor area.
Cut the connection.
Exactly. And what happened? The anti-PD-1 therapy suddenly worked much better. Significantly more effective tumor control. It was like removing a shield or a barrier that the nerves were providing, allowing the immunotherapy to actually get in and work. It strongly suggested those nerves were actively interfering. Then they did the flip side, the mirror image. In a different mouse model, they deliberately surgically injured the nerves near the tumor, a procedure called axotomy.
So they cause nerve injury on purpose.
Precisely. And just as they hypothesized, making the nerves injured made the anti-PD-1 therapy less effective. Tumors grew faster, control was worse. They even replicated this finding in humanized mice carrying human Seiki-C tumors, which really boosts the relevance for human treatment.
Okay, let me process that. Patient data shows more nerve injury in people who don't respond. Remove the nerves in mice, the treatment works better. Injure the nerves, the treatment works worse. What's the big takeaway from this part?
The big takeaway, really, is that these experiments provide strong, compelling evidence. Cancer-induced nerve injury isn't just a bystander effect. It's not just a symptom. It is a functional, active driver of resistance to anti-PD-1 therapy. The nerves are playing an active role, functionally involved in whether these crucial therapies succeed or fail. That's a fundamental shift.
A huge revelation, definitely. Okay, so we know nerve injury is bad for immunotherapy, but how are the cancer cells doing this damage? Is it just like squeezing the nerves or something more specific? And what actually happens to the nerves when they get attacked like this?
Yeah, it's much more direct and insidious than just squeezing.
Yeah.
It's a physical attack. They used co-cultures, basically growing cancer cells and nerve cells together in a dish, and they watched. Within days, the cancer cells made direct contact with and invaded the nerve axons. And this led to visible myelin breakdown. Myelin is that fatty protective sheath around nerves, right? Like insulation on a wire. It's essential for fast nerve signals.
Right.
Exactly. And they used electron microscopy, which gives these incredible zoom-in views, and confirmed it. They could see the layers of the myelin sheath, the lamellae getting all tangled and just disappearing where the cancer cell touching the nerve. It's literally stripping the insulation off, leaving the nerve exposed, vulnerable, dysfunctional. And importantly, they looked for synapses, the usual communication points between cells. There was no evidence of that. This wasn't the cancer trying to talk to the nerve in a normal way. It was just physical destruction. Sabotage, really.
So it's not just invasion, it's active destruction of that protective myelin layer. Did they see functional consequences? Did the nerves stop working properly?
Oh, absolutely. Functionally, it was clear-cut. They did electrical conduction studies on the skin of mice with these tumors. And they found nerve conduction speeds were significantly lower, and the electrical patterns were abnormal. Exactly what you'd expect if the myelin's degraded. The signals just weren't transmitting properly.
Like a faulty wire.
Precisely. And crucially, to bring it back to humans, they validated this in patient samples too. They found a clear link between injured nerves in patients and the presence of something called degraded myelin basic protein, DMBP. DMBP is basically the molecular junk left behind when myelin gets chewed up. So it confirms this destructive mechanism is happening in people undergoing treatment too.
Okay, so it's like peeling the insulation off a wire, but nerves aren't just passive wires, are they? They react. What happens inside the neuron itself when it gets injured like this? What's its response?
And that's where the story takes another really surprising turn. The injured neurons, they don't just sit there passively. They fight back, they react strongly. But ironically, their reaction signal actually ends up being bad for the overall anti-cancer immune response. They saw huge expression of that ATF3 gene we mentioned earlier, the nerve's panic button. That's a known injury response marker. But here's the kicker. They did RNA sequencing on these injured neurons, looking at all the genes being turned on. And what popped up? A massive upregulation of immune-related pathways. Things like TNF signaling, type I interferon signaling, IF0, IF-MEL, IF-NES signaling, and especially, really prominently, IL-6 signaling.
Wait, the neurons are activating immune pathways.
Exactly. That was a critical and frankly unexpected finding. The neurons themselves were kicking off this strong inflammatory response. And it wasn't primarily the cancer cells pumping out IL-6 in this specific context. It was the injured neurons. They were the source.
So let me get this straight. Cancer cells attack, destroy the myelin. The nerve gets injured. Then the injured nerve itself sends out inflammatory signals like IL-6, which then messes up the immunotherapy. What's the bigger picture meaning here?
The bigger picture is this really vicious cycle they uncovered. Step 1, cancer degrades myelin, causes nerve injury. Step 2, the injured neuron reacts autonomously by launching this inflammatory cascade with IL-6 and other signals. Step 3, turns out this specific inflammatory cascade isn't helpful. It actually creates exactly the kind of environment that promotes resistance to anti-PD-1 therapy. It's a self-reinforcing loop that helps the tumor protect itself.
Wow, it's like the cancer tricks the nerves into becoming unwilling accomplices in its defense strategy. So this nerve-driven inflammation, is it just a tiny local problem right around the nerve, or does it spread out? Does it affect the whole tumor?
Great question. And no, it's not just local. It ripples outwards, creating a hostile environment throughout the whole tumor, a perfect storm for resistance. They saw in the mouse models, as the tumors grew bigger, more nerves got injured, and this correlated directly with an increase in immunosuppressive macrophages, immune cells that shut down attacks and exhausted CD8 plus T cells, the killer cells that are too worn out to fight, spreading throughout the entire tumor microenvironment, the TME.
So the bad guys increase everywhere.
Pretty much. And then they looked in patient samples again from CSCC, and also pancreatic cancer, another cancer notorious for P&I. They found these injured nerves were actively attracting those immunosuppressive cells, creating these little perineural niches packed with cells that hinder the immune response. What's more, in the non-responders to anti-PD-1, the nerve injury markers directly correlated with immune suppression markers within that niche. It points to this chronic, sort of pro-healing inflammation that unfortunately works against killing the tumor. They even saw increased markers of neurodegeneration, like PARK-K7 and PNK1, in these niches and non-responders. Suggests the nerves aren't just injured, they're actively breaking down, probably fueling the fire even more.
But you said it spreads beyond the niche, that this local damage has wider influence. How did they show that?
Yeah, that's where some really cool technology comes in spatial transcriptomics. It lets you map out gene activity across a slice of tissue, see where things are happening. And it showed something remarkable. Areas in the tumor with lots of this CI9, this nerve injury signature, also showed co-localization of that whole immunosuppressive inflammation phenotype across the entire tumor section, not just immediately next to the nerves.
So the bad influence spreads far.
It seems so. The patterns of immunosuppressive cells found near the injured nerves correlated with similar patterns in areas quite far away from those nerves. It suggests that this chronic inflammation triggered by nerve injury kind of extends its reach. It skews the whole immune climate in the tumor towards suppression and T-cell exhaustion. And that becomes a major barrier blocking the anti-PD-1 therapy from working effectively anywhere in the tumor.
Okay, this paints a pretty bleak picture for those patients, but it leads to the critical question. If nerve injury causes this resistance, can we do anything about it? Can we reverse it?
Yes. And this is where the research gets incredibly exciting and helpful. The answer seems to be, yes, we can intervene. They tested several strategies. First, they tried targeting the nerve's own injury signal. Remember ATF3, that panic button?
Yeah.
In melanoma mouse models, they used genetic tools to knock out ATF3, but only in the nociceptor neurons, the pain-sensing ones often affected first. And the results? The tumors were smaller. Not only that, they saw more active anti-tumor T cells, and the macrophages shifted towards an M1-like pro-inflammatory anti-tumor state. They became fighters again.
Just by stopping the nerve's distress signal.
Exactly. It showed that blocking the nerve's own response to being injured could actually make the immunotherapy work much better.
Okay, that's targeting the nerve itself. What about the downstream signals, those inflammatory molecules like IL-6, the nerves release? Can we just block those?
They looked at that too with really promising outcomes. Remember how the clinical data hinted that non-responders had high type I interferon signaling? Well, type I FNEs can be good acutely, but chronic signaling can actually suppress immunity. So in their mice with myelin damage, they knocked out the receptor for type II-AFN, IFNAR1-KO. And guess what? It restored the effectiveness of anti-PD-1 therapy. Blocking that chronic IFN signal reversed the resistance caused by nerve injury.
Wow.
And maybe even more directly relevant, they targeted IL-6. They combined standard anti-PD-1 therapy with an antibody that blocks the IL-6 receptor. This combination significantly reduced tumor viability in the mice with nerve demyelination. And crucially, this benefit was dependent on the demyelination. It confirmed IL-6 was playing a key role specifically in this CI9-driven inflammation, not just general tumor inflammation.
So multiple potential ways to break that cycle. Blocking the nerve's reaction, blocking the inflammatory signals. What's the big picture implication here?
The big picture is that these findings are incredibly encouraging. They show we can intervene. By targeting different points in this CI&I pathway, whether it's the nerve's initial ATF3 response or downstream signals like IFNI or IL-6, we can potentially reverse the immunosuppression caused by nerve damage. And by doing that, we can restore the effectiveness of anti-PD-1 therapy. It basically provides a whole new roadmap for developing combination strategies.
This really does feel like more than just adding one more piece to the cancer puzzle. It feels like it's revealing a whole new dimension. What makes this so innovative, would you say? What's the real paradigm shift here?
I think the central advance is this complete reframing. Like we discussed, nerve invasion or injury used to be seen as just a symptom, maybe a late-stage marker of a bad tumor. This study fundamentally repositions it as an upstream driver of treatment failure, specifically for immunotherapy. It pulls neurobiology out of the periphery and puts it right at the core of cancer immunology, establishing this critical nerve-tumor immune triad. The innovation is showing this isn't just proximity, it's functional communication. And importantly, if you interrupt that damaging communication by protecting the nerve, blocking its response or blocking its signals, you can actually restore sensitivity to therapy. That gives us a completely new angle of attack.
So translating this forward, what could this mean for actual cancer treatment for patients in the clinic?
The potential translational impact is huge. First, think biomarkers. We could potentially develop tests based on nerve injury looking for ATF3 in nerves, or maybe myelin breakdown products in biopsies, or even blood. These could help predict, maybe much earlier, which patients are likely to resist anti-PD-1 therapy because of this CNI mechanism. That allows for personalized treatment planning right from the start.
Tailoring the therapy.
Exactly. Second, and maybe most excitingly, it opens up this entirely new axis for combination therapies. We could pair standard anti-PD-1 drugs with treatments that specifically target this nerve-tumor interaction. Maybe drugs that protect nerves, pharmacologic neuroprotection. Maybe in very specific situations, targeted nerve denervation. Or perhaps more immediately, repurposing drugs that block those key inflammatory signals, like IL-6 inhibitors, which are already approved for things like arthritis. And because this mechanism is different from how PD-1, PD-L1 blockade works, it's what we call an orthogonal approach. It's complementary. It could genuinely boost efficacy for patients who currently get little or no benefit.
It's clearly a powerful foundation they've laid. What are the next big questions? What do researchers need to figure out next?
Oh, definitely. This is really the start of a whole new field, in a way. Key next steps include validating these findings across a much wider range of cancer types. Nerve biology differs between organs, so we need to see how general this is. We also need to think very carefully about delivery. If we're targeting nerves or nerve signals, we ideally want to do it locally within the tumor environment to avoid potential side effects throughout the body's nervous system. That requires developing sophisticated local delivery methods. And one more crucial point. We need to be sure that by blocking these injury signals, we aren't accidentally interfering with any beneficial roles nerves might play in fighting cancer. It's likely a complex balance. So lots more work to do to really understand the nuances and optimize these strategies safely and effectively.
What an absolutely fascinating deep dive. We started with this really tough mystery. Why don't some patients respond to our best immunotherapies? And we've uncovered this surprising culprit, the cancer-damaging nerves and potentially powerful solutions hidden within our own nervous system.
Indeed. Understanding how cancer can actually weaponize nerve injury to create immune resistance, it's a profound shift. It highlights that it's not just about the tumor cell or just the immune cell. It's about the intricate, complex conversation happening between the tumor, the immune system, and the nervous system, a whole new layer to consider.
So what does this all mean for you listening? Maybe, just maybe, the key to unlocking immunotherapy's full power for so many more patients lies right there in listening more closely to the whispers or perhaps the screams of our nerves. It could truly transform how we understand and how we fight cancer forever.